Hello friends, in this video we'll discuss about the proteins actin and myosin. First of all, we see the structure of actin. You see this beaded structure of filamentous beaded structure. This is one strand and this is the other strand. This is the actin filament. Now tropomyosin in this diagram, we can see, if we enlarge this picture, this gray colored filament, you can appreciate this gray colored filament growing here. This whole gray colored filament is tropomyosin. It is a regulatory protein. Now there is a troponin complex. You can see this purple colored structure. This is troponin complex. It is a set of additional regulatory proteins bound to actin filaments. Now, what happens when the motor neurons, they trigger the release of calcium into cytosol? Calcium binds to the troponin complex. See, this is the troponin complex, and the calcium which is released into the cytosol, it will bind to the troponin complex. What happens after its binding? Exposed myosin binding sites. What are myosin binding sites? You can see these black colored dots. These black colored dots are myosin binding sites. These myosin binding sites are actually covered by this tropomyosin. But when calcium is released, what it will do? It will bind to this troponin complex, and now it causes the troponin complex to change shape. Whose shape is changing? This troponin complex is changing shape, as a consequence of which this tropomyosin will expose these myosin binding sites. See, because of configuration in this troponin, tropomyosin has exposed these sites now. Now, dislodging of tropomyosin from myosin binding site occurs. Then what happens? Cross bridges. What are cross bridges? We all know that myosin is a racket-like structure. Head and a tail is the structure of myosin. This head will attach to these myosin binding sites, and the bridge which is formed is known as cross bridge. Cross bridges will be formed. This causes what? Sliding of filaments. And sliding of filaments will cause contraction of muscle. The reverse happens when what happens? Falling of calcium ion concentration is there. Then binding sites will be again covered and the contraction will stop. Arrival of action potential at synaptic terminal. What it causes? It causes release of acetylcholine. Now we look at this diagram first of all to appreciate this thing. See, this is a synaptic terminal. And this is the axon of motor neuron, right? And this is a single myofibril. Myofibril is again a myofibril. And what is this? Plasma membrane. This is the plasma membrane of muscle fiber. If we cut this plasma membrane, we see these T tubules. T tubules are infoldings of the plasma membrane, and after cutting we can see it like this. Now each myofibril is surrounded by sarcoplasmic reticulum. Around myofibril. It is surrounding the whole myofibril. In the next myofibril we have striped off this sarcoplasmic reticulum, so you can appreciate this myofibril now without the SR or sarcoplasmic reticulum. Now, what happens? Acetylcholine is released, and this will bind to receptor on the muscle fiber, and it will cause depolarization. Obviously, action potential will initiate. It will spread to the T tubules of the plasma membrane. What are T tubules? Again, enfoldings of plasma membrane. These T tubules are in close contact with the sarcoplasmic reticulum. Here we can very well see sarcoplasmic reticulum is here, and these are the T tubules. They are in close contact with each other. Now, action potential will spread along the T tubules, and it will trigger changes in sarcoplasmic reticulum. What will happen? Opening of calcium channels. Calcium stored in sarcoplasmic reticulum will flow through open channels into the cytosol. It will go into the cytosol. See here, this is the calcium. Calcium released from sarcoplasmic reticulum. This is calcium. It was released into the sarcoplasmic reticulum. It will bind to the troponin complex as we studied just now, and the muscle fiber contraction will start. When the motor neuron input will stop, filaments will slide back, muscle will relax as proteins in the sarcoplasmic reticulum pump calcium back to the starting position. Myosin binding sites are blocked again. Right? So this is how the contraction starts. Now, what is tetany? Sometimes there is a high rate of muscle contraction, so they do not get time to relax in between. Muscle will keep on contracting. So such a continuous state of contraction without relaxation is known as tetany. There is also a disease called tetanus. In this disease, what happens? The muscle will keep on contracting, and it can be sometimes fatal for a person. Now we'll discuss about the cardiac and the smooth muscles. Cardiac muscles are striated. They initiate contraction without nervous system input. Why? Because they have pacemakers in them. Pacemakers will do this job. Cardiac muscles also have intercalated discs. What are intercalated discs? They are specialized regions who will compile the signals. Action potential lasts 20 times longer than the skeletal muscles and cardiac muscles. Now, when it will last so long, it won't harm the cardiac muscles. Why? Because the refractory period is long. Refractory period is the resting period between contractions. So, no tetanus or no summation occurs. What is summation? When there are two constant contractions, one contraction may sometimes get summed up with the other. This is known as summation, but it won't occur because the refractory period is long. Now, what are smooth muscles? They are present in our viscera. Viscera means what? Are smooth the intestines, the stomach, the fallopian tubes. These are all the viscera. Smooth muscles are also present in the arteries, etc. And they lack striations. Why they lack striations? Because actin and myosin are not arranged regularly in these smooth muscles. Now, we also have to study the structure of myosin, which we haven't yet studied. So let us have a look at the structure of myosin. See, the structure of myosin is like this. This is the tail. This is also known as, and this is the head. Then this head has one place here. These are the ATP binding sites. ATP binding sites. Now in the tail there are actin binding sites. Now this is actually a myosin monomer. This is also known as light meromyosin. Light meromyosin. And the head part is also known as heavy meromyosin. This is the structure of myosin. Thank you.